get started today by firstly revising our sort of big ideas from Module 6. So we won't go through obviously the whole Module 6, but we will be revising the big ideas and the key concepts so that we can then go on and spend a lot more time going through Module 7. So, as you would hopefully by now be familiar with it, this is what the HSC syllabus for biology looks like. You are studying effectively 4 topics. The first one is heredity, followed by genetic change, infectious diseases, and non-infectious disease and disorders. So, since generally most schools sort of stick in that order of Module 5 to 8, I'm assuming that most of you have done Module 5 and 6 and are now going to start 7, or like that would probably be the majority of us. If you haven't, if today is your first time having a go at both Module 6 and 7, in case the school has done 5 and 8, just try to follow through the best you can. Please ask lots of questions and I'll try and break everything down so that we to those of you who have done it, you are building on your knowledge, and to those who haven't yet had a chance to do it, you can again start to build our foundational knowledge that you can take along with you into term 2, term 3 for year 12. So let's get started. So we're starting off by firstly looking at Module 6. So the first dot point of Module 6 is asking us to look at mutagens. What are the different types of mutagens and what mutations do they cause? So, we look at what mutations they cause in the next slide, but that's sort of the first big point. What are mutagens? So a mutagen is just gonna sneeze. Okay, it's been happening all day. I feel like I need to sneeze, and then I think about it and it just goes away. So apologies for that. But as I was saying, so a mutagen is an organism that causes mutations. A mutagen can also be known as a mutant. So there are 3 types of mutagens that we're looking at, including chemical, electromagnetic, and naturally occurring. So chemical mutagens include radioactive agents like uranium, other chemicals, and it causes structural changes. We've got metals, including arsenic and nickel, that can affect the DNA replication process. And finally, we've got intercalating agents like ethidium bromide, which can cause frame shift mutations. And we'll have a look at what frame shift mutations are in just 1 second. There are also electromagnetic radiations. Those are like the most prominent ones. They happen, they're high energy and they are highly penetrative. They alter DNA bonding, which causes the rearrangement of the structure, and now we know that because of the processes that are occurring in the DNA, they can have a huge impact on the genome, and as a result cause mutations. An example of an electromagnetic radiation mutant is UV light. So radiation from the sun can be super harmful in high doses, and as a result it can cause pyrimidine dimers. So that is when you would have the bases sort of, and actually there's an image here. I was trying to explain it with my hand, but I'm pretty sure I've got an image here for us. So this is the electromagnetic spectrum, and we can see that in terms of electromagnetic radiation, we would know that gamma rays, so it goes from radio to gamma, and this side as you go along it becomes more penetrative. So that's that. Okay, moving on. So now here we go. These are our pyrimidine dimers. So we've got, as you can see, so what happens as a result of radiation to the DNA. So there can be hydrogen bondage breakage. So by now, as we would know that the bonds are joined, so the bases they have hydrogen bonds holding them together. So hydrogen bonds here can break. They can also be pyrimidine dimers, so this is when you can see the bases, they turn inwards to connect to one another instead of connecting straight across. There can be a loss in bases, base change, cross linkage can also happen. There can be single strand break and also protein cross linkage. So there are lots of things that can happen here. I would say to really know pyrimidine dimers, what that is, how that happens. and of course hydrogen bond breakage. And yeah, those are the sort of the big ones that we need to know because they have a huge role, in changing the structure of the DNA. Okay, moving on now to inquiry question 2. So how does mutation introduce new alleles into a population? So firstly, as you can see here, these are firstly the results of mutagens. So it can have a huge impact on phenotypic expression. And as you can see here, we've got ionizing radiation that has affected the DNA. So we've got the X-rays or gamma rays over here breaking the single strand and also breaking the double strand. On the other hand, we've got non-ionizing radiation, which causes pyrimidine dimers to form between thymine. So it can form between thymine and guanine. Yes, it's always good to remember that. But this is an example here of thymine dimers. So these are forming between two thymine bases. So that's that. Alright, let's have a look now at what happens as a result of these mutations. So we've got point mutations. So point mutations are a type of mutations that happen as a result of damage to DNA, and there are two sort of distinct categories for them. The first one is substitution, so they can be substitution mutations or they can be frameshift mutations. So with frameshift mutations, there are two specific types. There is insertion or deletion. So we've basically got the three types of mutations that happen as a result of these and the three effects. So the three types they can be silent mutations. So silent mutations is when there is no effect on the codon, on the DNA. So that there's no effect, it's silent, it doesn't have any effect on the DNA. The second is missense, which is when we see a change in the amino acid because the codons have been disturbed. There has been a change in the codons. Finally, we've got a nonsense mutation, which means that a stop codon has been prematurely introduced, which is going to affect it is going to prematurely stop the process and affect the DNA that is being produced. Sorry, that is going to affect the proteins and the peptide synthesis that's happening. So those are firstly point mutations. Now we've got chromosomal mutations. Now chromosomal mutations is when actually, let me just quickly go back just because I realized I forgot to mention something. So with point mutations, that's the big category. So there are point mutations, but there are two types of point mutations. There's substitution, when a base has been switched out, and then there's framework, when either a base has been inserted or it has been deleted. And so as a result, you get to see three different types of effects. Three different types of changes, and then there's specific effects. So just wanted to flag that out. Okay, let's have a look now at chromosomal mutations. So chromosomal mutations usually occur as a result of errors that happen during meiosis, and crossing over. And that could be because crossing over occurs incorrectly. So there can be chunks of DNA that was screwed up, and that can cause structural mutations to happen. We can also see sister chromatids being incorrectly separated. So during anaphase, when the chromosomes are supposed to separate, the DNA it may not be separated properly, which can then again cause a number of mutations as well. Sorry, let me just check my charger's plugged in. Just sparkling. All good. Alrighty, just making sure. Yep. Okay, perfect. So let's now have a look at the effects of it. The effects are usually severe because now we've got issues within the DNA happening at a bigger level, like it's happened during meiosis, and it is going to affect the phenotypic expression, but it's also going to affect their gametes. In some cases, it can affect their gametes as well, depending on what the mutation is. So there are 4 types of structural chromosomal mutations. They include deletion. So deletion is when a chunk of that chromosome has been deleted, as suggested by the name. There is inversion, when a part of that DNA has been inverted inside out. We also have translocation, when a part of the DNA has been misplaced. The chromosome has been displaced. And finally, we have duplication, where a part of the chromosome has been duplicated. So those are the 4 types of structural mutations. And if we've got mutations in the number of chromosomes, which can again happen if we've seen if it's happening during meiosis and it affects the separation of the strands, there can be mutations in the number of chromosomes. And there are 2 main types: there's aneuploidy and polyploidy. So aneuploidy is when the overall number of chromosomes of the offspring differs to that of the parents. So, for example, in Down syndrome, the individual has 47 chromosomes instead of the usual 46 chromosomes, and that is because they have 3 copies of chromosome number 21. So that's aneuploidy. Then we've got polyploidy, which is when an organism contains more than 2 sets of homologous chromosomes. So, for example, triploidy is a condition in which the fetus has 3 copies of every homologous chromosome instead of 2 copies. And that is obviously going to have a severe effect, on a large number of genes. So, we've got, so as I've introduced you earlier to TuteSpot, here are some examples of, for example, little guides to looking at mutations, and these and I tutor biology here at TuteSpot. I tutor biology, PDH, PE, and English Advanced, and this is one of the examples of how we like to deliver our content. Obviously, we have a very similar sort of slide, not slide structure. Sorry, we have a very similar structure in terms of go through the slides, but some of the resources and the way in which we present our content includes like infographics like this, which can be so helpful in visualizing what is happening, and these are here for you if you want to have a look and use them and add on to your notes as well. Okay, now let's have a look at the second big dot point, which is biotechnology. So with biotechnology, our inquiry question is, how do genetic technologies affect Earth's biodiversity? So this is something that you will come back to, that you have obviously looked at briefly in Module 5, and you will come back to in Module 7 and 8 as well. Like, you will come back to this idea of biotechnology, and we are going to go through Module 7 in a while, so you'll sort of see how that ties up with Module 6. So there are 3 different types of uses of biotechnology that we are looking at. Firstly, we're looking at the use of biotechnology in health, so medical biotechnology. And examples of that include, of course, biofarms, but those are not just the only one. And the big examples, of biotech, of medical biotech, is vaccines and antibiotics. Having now, gone through the pandemic, we are all very aware of vaccines and, antivirals and stuff, and how they've played a huge role in curbing the pandemic. There are also stem cell treatments that are being developed, and that have some have been developed, some are being developed, and there are changes being made, and that includes skin grafts, treatment of cancers, and autoimmune diseases. We also have industrial biotech. So some examples include biocompatible materials like biopolymers derived and also produced by bacteria or plants, and as well as biodegradable plastics. Living in a world where we are currently dealing with the effects of climate change, along with obviously many other issues, biodegradable plastics will have will play a huge role in the way in which our world functions. So that is again something that is being constantly researched and improved. We also have the use of enzyme biotechnologies and processes like fermentation and food production that once again play a huge role in making industrial processes more efficient. Finally, we've got environmental biotechnology, and we will again be talking about all of them in a lot more depth. But I think environmental biotech is one of those that we sort of know, like we use every day, but we don't really think about that as like biotechnology, right? So we've got bioremediation, which is development of microorganisms to help clean up pollution. And we also have the use of genetically modified crops and organisms in agriculture that is being used to better, suit a changing environment. And that is something, for example, BT cotton, corn, bananas, Cavendish bananas, all genetically modified. So it is something that we obviously that we interact with every day, use every day, but we may not actually consider it biotech. So here are the three different types of genetic technologies that we will be looking at in this module. You will see that in the syllabus some of them are specified, so the ones that I've highlighted in pink there for you, those are the ones that are specified in syllabus, but of course you still want to know examples of the others as well. So we've got reproductive technologies including artificial insemination, which is used in the livestock industry. We've got in vitro fertilization, so IVF, which is a fertility treatment, and finally we've got artificial pollination, which is used in agriculture. So these three are reproductive technologies. Then we've got cloning techniques. And cloning techniques include firstly whole organism cloning, so it's used widely in livestock industry. I think by now we would all know Dolly the sheep. So Dolly the sheep is an example of whole organism cloning. There are many other out there now. Another one is Injaz the camel. That was a camel in the Middle East that was cloned. We also have therapeutic cloning, which is happening in medicine, again a field that is still being researched and explored. Finally, we've got gene cloning, which is happening in both medicine and industry. So like I said, the ones in pink are the ones that are highlighted in the syllabus, so you definitely need to know those. And the ones in blue, of course, are extra examples that I still think you should know, just in case you get a question where you don't want to keep talking about one thing and you can bring in other relevant examples. And the syllabus also says including but not limited to, so it's a good idea to have some extra examples up your sleeves. And finally, we've got recombinant DNA technology. So that includes gene sequencing, which is used in medicine and research. It includes transgenesis, which is also used in agriculture and biotech. Gene therapy, ELISA, and CRISPR, which is used in medicine and is a molecular tool. So here I've got an example for us. So we've gone through the content, and now let's have a go at adapting it to this specific question, which is from the 2019 paper. The application of reproductive technologies in plant and animal breeding limits genetic diversity. So to what statement is this statement correct? A question like this, to what extent you need to specify what your opinion is. You have to say to a significant extent this is true, or this is not true, or this is untrue to a great extent. Something like that, where you actually answering the question. And with a question like this, you need to be able to, there's a lot that you can talk about. So structure is crucial. It is worth 6 marks, so that's very important. So structure is crucial, and organizing your points to sort of come back to this big idea. Almost think a bit like an English essay, you know. You want all your evidence to link back to your main thesis, to your main contention, your main argument. So kind of going through that structure. So let's not be scared. Let's now have a go at how we would structure this. So if we've got a question like this, I would obviously want to have an introduction that gives me definition of, reproductive technologies and how they are being used in plant and animal breeding, and evaluation, right? So that is the part about how they're being used. Evaluate it for me, and through that answer, to what extent do you think this is true? Then in your body paragraphs, you are giving me examples and arguments to link back to your main contention and prove that, hey, you are right. And here I might talk about 2 specific technologies. So for each one, I will define what they are, describe the give a brief outline of how they work, an example, and then comment on does it limit genetic diversity. So I have to give examples of reproductive technology, because that is what the question is asking me to do. The question wants to know if the application of reproductive technologies in plant and animal breeding limits genetic diversity. So I need to give examples to back my point up here. And finally, we want a conclusion that ties everything together and reinforces your evaluation. So here you link back to your body paragraphs. Again, like in English, you're signposting your themes. That is kind of what you're doing here. So that is that. Let us now move on to module 7.